Well, hello and welcome to the lecture. This is our lecture buddy for the moment. Hello, lecture buddy. Oh, Kung Fu. Anyway, we're going to be talking about organic chemistry with this YouTube lecture. So, simply put, we'll start with the basics. What is organic chemistry? That's pretty simple. All something needs to be organic is carbon, right? So if you have one or more carbon and the right arrangement, you are an organic compound. There are compounds that have carbon and are not considered organic. For instance, carbon dioxide, carbon double bonded with two oxygens, that is an inorganic substance, even though it's got carbon. So there's some various rules, but you don't really have to worry about those until OChem. Right, little buddy? Right. We're just going to point that out. Okay. Carbon is, of course, considered the backbone of all life on Earth. And we'll talk a little bit about why it is so versatile and so capable of providing all the chemicals required for life. But needless to say, we are carbon-based life forms. So let's get right into organic compounds. They can be very simple. C2H6 is an organic compound. So you draw, let's see, when we do drawings of organic compounds, we'll often do little lines like this. Ugh, bad with the mouse. Anyway, each one of these things right here, each peak, each point is considered a carbon. So, how many carbons I got here? I got, oh, so lazy. All right, so these are all carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9 carbons in this compound. Another thing about organic chemistry is that if I were to just draw this up, it is absolutely assumed that this has carbons with completed valences, and those valences are all completed with hydrogen, right? So just that line implies this hydrogen here. And it implies that there are two hydrogen coming off of here. So if you see just a line with little peaks there, just this, right? This has 1, 2, 3, 4, 5, 6, 7. So if I were just to show this, this would be a 7-carbon compound, and it would be completed with hydrogens, saturated with hydrogens. So C2H6 is pretty simple, right? You just have 2 carbons and then hydrogens attached. So there's a carbon, 2, 3, 1, 2, 3, 6 hydrogens, 3 on each carbon to complete the valences. And so here's a nice little example of C2H6, and they can be extremely complicated, like my little friend here. And a complicated carbon compound would be proteins. So this is just a quick example of proteins here, in which we have all sorts of compounds attached to here. So the light blue color is just our carbon, right? So these here are the carbon compounds, or the carbons. White is hydrogen, right here. Right? Blue is nitrogen, the dark blue. Red is oxygen. Gold is phosphorus. So you can see these are incredibly complicated compounds. These are always consisting of a chain or a ring structure, or both. So it can be a simple chain like I've been drawing. It can also have rings of interconnected carbons, and it can have chains with rings. They always have hydrogen attached to them. Organic compounds always have carbon and hydrogen. And you will often find a lot of those electronegative atoms attached to it, like oxygen and nitrogen, phosphorus, all over in there. So, 99% of any living cell of any type of living organism, whether it's my friend here or me, is made of hydrogen, oxygen, nitrogen, carbon, phosphorus, and sulfur. And it's just the arrangement of our carbon compounds that determines what we become. So that's pretty sweet. And that's one of the reasons why organic chemistry is so awesome. Just differences in the chains produce differences in the cell or the organism. All right, so we should talk briefly about the hypothesis of abiogenesis. So when we talk about how life may have begun on Earth, we talk about abiogenesis, which is life from the non-living components in the early Earth. So it's all about the origin of life on the early Earth. And on the early Earth, the source of our atmosphere was volcanoes. Outgassing from volcanoes, the gases that are thrown out when a volcano erupts, is what produced Earth's first atmosphere. And the question people who are investigating abiogenesis ask is, well, one, could life have come from simpler non-living ingredients? But two, before you can even get to the question of life is, can you get organic compounds from simpler inorganic compounds? So the key would be to try and replicate as many conditions as possible for early Earth. And of course, that would be water in the early oceans, water in the atmosphere, carbon dioxide, lots of carbon dioxide in early Earth, but that atmosphere being caused by the outgassing of volcanoes also led to some really poisonous crap being part of the atmosphere, like sulfur dioxide, carbon monoxide, and the rest. No, this is not a bullet list you are needing to memorize. I'm just showing you that we had a whole lot of inorganic gases in our atmosphere. Disulfur, chlorine gas. Remember I talked about how chlorine gas will tear organic stuff apart to fill its valence? Nitrogen gas, hydrogen gas, ammonia, methane. These are all inorganic compounds that were in the atmosphere. We know they were in the first atmosphere because the gases that volcanoes produce don't really change over time. So we just stick a probe down in a volcano and we measure what gases come out. And we can even measure the proportions of gases coming out of volcanoes. And then we can make estimates on how that atmosphere looked. So the very first experiment, I think, was started in 1954. Whoa, what just happened? And there have been 10 plus of these experiments. They're all variations on this basic theme, wherein we have two flasks. So we have a little flask here and a flask over here, and they are connected together, right? Okay, so then we have our connection up y'all, and it comes over here. pretty simple. These are designed to be a completely closed system. Once the experiment starts, there is no gas, nothing can get in and nothing can get out. It will only ever run as a closed system. So, I need to get this a little bit better. So, yeah, the whole idea is to trap a whole lot of gases and a bit of water in this system, and then see what happens. So we will go ahead and put some water in this lower flask here, right here, and we'll also add some fire. Oh, burny fire, burny fire, right? And that causes that water to evaporate. So we get water vapor evaporating through the system, right? And it comes over to this flask over here, which was the atmosphere flask. And we get water vapor into the atmosphere, where we have that huge mix of just generally awful gases like chlorine and disulfur and whatnot. In the atmosphere glass, we have electrodes, right? And these electrodes will periodically shoot electricity. Zap, 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 zap. And that electricity represents lightning in the early atmosphere, right? So you pass the water vapor through the atmospheric thing, and we simulate lightning in there, and then it rains down. We have some condensers that cool it enough that it turns back into water, flows back into our little ocean, and we see what develops there. So let's just take a look. We have that loop and two flasks with the ocean flask and the atmosphere flask. And there's the electrode in the atmosphere flask simulating lightning. We have a heat source to cause evaporation to go to the atmosphere flask. The water vapor is then interacts with the lightning and the early atmosphere. And we get rain, right? So, and then you notice on the drawing down there at the bottom, it has a sampling probe, which is a spot where you can insert a little probe into a port that will detect chemicals in there without actually entering the system. Because remember, this is a completely enclosed system. Once you start it up, there's no opening it, so there's no chance of contamination. So, oh my goodness. The question then is, what are the products of these 10 or more experiments, right? They've all been variations on a theme. Basically, you had the original, which is pretty much what we had the diagram and picture of. And then the others all tried to control for various other properties of early Earth's atmosphere. And essentially what we got were organic compounds. Loads of organic compounds, including the ingredients for DNA, like nucleotides. So we got amino acids, which are the building blocks of proteins. And we got sugars, which are important in energy production. And even more other organic compounds that I don't need to list off. So the question then, now that we know that in a simple experiment with early atmosphere, showing a variety of organic compounds forming, is this how life formed on our planet? Well, unfortunately, all we can say for certain is that with regards to abiogenesis, it is completely possible to form a large number of organic compounds from non-organic sources, including some very complicated organic compounds. So we can certainly get all the necessary organic ingredients for life, but can we show that life definitely formed this way? We cannot show that yet. There's been no evolution of living things in our experiments yet. But remember, Earth was going for like 500 million years before life appeared. Well, at least the earliest fossil record of living cells that we've found. And so we've been running these experiments for 70 years, 80 years maybe, no, 70 years, and we've already found all these organic compounds. So I kind of think that their findings lend support to the hypothesis that life developed on the planet from inorganic components, abiogenesis. So we cannot show that these experiments cannot show that this is how life originated on Earth, but it's pretty compelling. And a cool thing about this is there have been studies of old genes, right? So what is an old gene? If you look at the family trees of various organisms, right? Maybe you have humans, Homo sapiens over here, and then you have other apes and other primates and stuff, and it goes down, and then you have all the mammals, right? And we go further, and we get the other vertebrates, and you go further down the evolutionary tree, and you're getting all the evolved organisms. You got the plants maybe over here, right? You got a whole vast array of protists, right? Down here, just tons of them. You have your bacteria, and you have your archaea, right? The big family tree. And when we talk about old genes, what we're looking at is this sort of like convergence point here. We're trying to find the genes that the earliest common ancestor to all living things may have had. And so what we simply do is look for genes that are present up here, here, present in the protists, present in the archaeans, the bacteria, the plants. We look for genes that are present in almost all living things on the planet. And we consider those genes the old genes, right? They're old because if all the living things have these genes, then that means the oldest ancestor had the genes, and all the evolutionary lineages produced from those ancestors carried that gene with them. So an old gene is an ancient gene, a holdover from our evolutionary past. So these old genes that we've studied, we've been able to sequence some of them and figure out what they do. And what we find is that the products the old genes produce use the organic compounds we find in those experiments, right? Those experiments, those flask experiments produce a variety of organic compounds that you really don't see being formed normally right now, because those were from a volcanic atmosphere. So that volcanic atmosphere allowed for a lot of different kinds of amino acids that aren't all that common in the environment anymore. But these old genes use the components from those uncommon amino acids. So it's just kind of fascinating. It implies that the earliest life used a lot of the organic compounds that we found in those experiments. So, pretty cool, isn't it, buddy? Is it cool? High five? No? No? Okay. Hey! I won't point at you again, you little whiny bugger. Okay. So just to drive home the interesting bits of organic chemistry and amino acids, which are the basis for all proteins, right? The question becomes, how rare are amino acids? If we can produce them from a simple experiment with just 2 flasks and a really, really simple estimation of Earth's atmosphere, with a whole lot of factors from our planet not replicated in the flask system, just how rare are amino acids? Well, one thing I always love to point out is the Murchison meteorite. It fell down to the planet and landed in Australia, right? And it landed intact enough, like it didn't explode, so that they were able to take a core sample. So they took the meteor, put it in sterile conditions, and using sterile tools drilled in and got a core sample from the center of this meteorite. Hypothetically, this would be ore and various stuff in the center of the meteorite, which has been untouched by Earth's atmosphere, uncontaminated, and a representation of what is on a lot of these meteors flying through space. And the answer of what's in that is that there were a large number of amino acids in there, right? So it was actually pretty amazing that in our own galaxy, traveling between the stars amongst meteorites, are these, rocks full of amino acids, right? And in early Earth's atmosphere, it was very thin, so these meteors were bombarding the planet constantly. So who knows? Perhaps some of life is alien in nature. Perhaps some of the amino acids that jump-started life came from space. My little buddy here certainly looks like he came from space. All right. So, was it abiogenesis? No. Well, we can't say for certain right now, right? We have not yet been able to replicate abiogenesis. However, there is a ton of circumstantial evidence that I find extremely compelling. All right. On to carbon compounds. So, carbon being the backbone of life, it's worth noting some of the reasons why carbon is so versatile and so effective at being a component of living things. So here's a carbon atom. It is atomic number 6. So how many in the valence? Well, inner orbital has how many? 2. So the valence has 4, which means how many single bonds could it make? Here's its nice little valence, and the answer is, of course, 4. It could make potentially 4 single bonds. And what's cool about the carbon single bonds is that they will occur at angles, right? So a carbon compound, right, will have bonds, single bonds forming at these little predictable angles. So maybe these are little hydrogens. So there's a hydrogen, there's a hydrogen, there's a hydrogen, and there's, sure, why not, a big one. And they form at these angles, and they call it a tetrahedron or a 3-sided pyramid, right? So, one side, there we go, bop, and bop. Crappy drawing, but you can see there's 1, 2, 3 sides. Easier to look at the shadow. 1, 2, 3 sides to this pyramid. However, when you form a carbon double bond, say just this carbon-carbon, it will alter the geometry. So instead of having these angles, you suddenly get linearity. So you can see we don't have as much of an angle anymore. We'll still have a little bit of an angle coming off either carbon here, but not much. We certainly don't see that tetrahedron anymore. So different kinds of bonding, different sorts of covalent bonds will directly change the geometry of these carbon compounds. But anyway, because each carbon can load up 4 single bonds, we can produce extremely long chains full of different atoms, causing all sorts of different properties to show up, right? So here's an oxygen here, and there's an oxygen and a hydrogen, right? They call that a carboxyl group. Then we'll just put over here a little nitrogen and 2 hydrogens, and that's an NH2. And so when we have an NH2 or amino group and a COOH or carboxyl group or carboxylic acid, this becomes an amino acid. Woo! So you can put all sorts of different stuff on these carbon chains, and you can alter whether or not they're hydrophobic or hydrophilic. You can add polarity to certain parts. So you can get a lot of variation in these things. So carbon is extremely useful when it comes to building complicated, large molecules. Molecules we call macromolecules. So macromolecules are able to form simply because carbon is so versatile. So these bonds form at these angles. When they're single bonds, they're all 109.5 degrees, not a number you have to memorize, but just that it forms that cool 3-sided pyramid, the tetrahedron. And then double and triple bonds add linearity to it. And so we can get all these neat things coming off of it. We can get the rings and the chains and all these fascinating, large, complicated carbon compounds. So, very, very cool. So, continuing on to the shape of carbon compounds. So you might see the word conformation here and there in my lectures throughout the semester. Conformation is a fancy word. If you're into like dog or horse shows, you might actually know it. It basically means how it's put together, or its 3-dimensional shape, right? Because I could draw a circle or whatever that is, and that's a shape, but it's not conformation. That's shape, not conformation, because conformation is the 3-dimensional shape of a structure. So, carbon comes in a large variety of conformations. Rings, chains, and different things you add to these change all the properties, and it produces that massive diversity. Let's talk about a neat thing we find in a lot of organic compounds: isomers. So an isomer is basically where we have the same chemical formula, but there are different ways it can be put together, and that can produce different properties. So we have identical elements, identical atoms in the compound, but they're put together in slightly different ways. And that can alter the properties of the carbon compound. So first up is structural isomers that differ in covalent arrangement. What that means is Though it still has all the exact same elements, each isomer is slightly different based on where those elements show up on the compound. Here's a nice example of a benzene ring with a carbon-chlorine bond coming up off of it. So we can see pretty simply right here, we've got CH2Cl, but then if that Cl moves, changes its covalent arrangement, now we have a CH3 and a Cl, and we're still identical elements, right? Because our Cl moved here, we lost an H, but now it's showing up here. So, and that chlorine can move all along here and produce different kinds of structural isomers. So, structural isomers are neat, a pretty simple type of isomer. They're just differing in where the little parts are on the compound. Same exact chemical formula, made of the same elements, just different placement depending on what kind of isomer it is. And so we could actually express these isomers with numbering, right? So we might name this carbon 1, then we have 2, 3, 4, 5, 6, and 7. I think I named this incorrectly because it's been forever. I think maybe this one is supposed to be 1, but we'll just go with it because I'm just illustrating, right? So now 1, 2, 3, 4, 5, 6, 7. That's as far as I need to go to illustrate this. All right, so here we'd have 1 chlorine, and then like septa something or other to imply the ring. Septa for 7 carbons, and something about the ring. And then over here we'd have, 3 chlorine, whatever. So we can identify an isomer because we'd have the same chemical formula, but if we put just the 1 or the 3, we'll know which carbon that chlorine is coming off of. So that's how we can identify different structural isomers. Cis-trans isomers are a fascinating sort of isomer, and these have to do with double bonds in a chain. So, double bonds in a chain. See, single bonds will rotate around, kind of like, let's see, so if we have our carbon, and we have our hydrogens, right, and we have another carbon, we have some hydrogens, we'll just make C2H6, right? These carbons can rotate like this, and then each of these hydrogens can sort of rotate, and these carbons can rotate in different directions. One could rotate this way while the other rotates this way. So one rotates along here while the other's rotating along here. So they can rotate in opposite directions. Each of the hydrogens can rotate, and believe it or not, the rotations can actually change the properties of the compound. But when you add a double bond, right, this essentially becomes an acyl. These carbons cannot rotate in opposite directions anymore. They are forever rotating in the same direction now. So our hydrogens might be able to spin around a little bit, but that won't affect much anymore because we've got this big acyl here. So when you have a double bond, you impede the ability of these atoms to rotate in their compound. And when you cease rotation, right, arrangement around the double bond becomes more important, right? Because if I've got a carbon here and a carbon here and an oxygen here and an oxygen here, and I don't know, we'll throw in a couple hydrogens there, right? So this carbon could rotate and this carbon can rotate, and basically as it rotates around, this oxygen could end up, you know, anywhere really. As the carbon rotates, the oxygen might rotate around. And so basically these atoms in the bond, they'll rotate until they find the most sort of like energetically and polarity-wise sort of most stable energy state. So as they rotate. But once you get this double bond and you have some oxygens, they don't rotate. So you can actually like classify the shape of these, not the shape, but the arrangement of these atoms around the double bond. So cis-trans is sort of stating, I got you, pal, come on. Cis-trans is basically talking about how those atoms are arranged around that non-free rotating double bond. Because now it's actually important what side these oxygens are, right? Because if these oxygens are on the same side here, each one is electronegative, and we're actually creating a polar region, right? We're going to have polarity on this side, and we're not going to have that electronegative region on this side. So, whereas if our oxygens are on opposite sides, here, carbon, carbon, two, right? Here's an oxygen. Here's an oxygen. Hydrogen, hydrogen. Right, now we don't have a real distinct side. You know, there's something electronegative here, but just on the opposing side, there's something electronegative. And so you don't really get a distinct polarity around this double bond. One side of the double bond isn't polar versus the other. So, and these are two basic arrangements. Now, cis-trans is not all about polarity. Polarity is just a convenient way to illustrate it. So, a cis isomer with my oxygen example right here, this is a cis isomer, and a trans isomer is where the oxygens sort of, you could think of it as transmit across. Trans across the double bond there. So cis, what we're referencing is on the same side. Trans, they're moving across. So you see these sometimes in fats too, right? So a fat is a big long carbon compound, and it's a big chain. So like here's a simple fat, and if I don't put anything on here, that means it's all carbon and hydrogen. And that would be a saturated fat, because every carbon is saturated with the maximum amount of hydrogens it could possibly have. Whereas if I add a double bond, like this, it's no longer saturated. This is unsaturated. Oh, this is looking great. But more importantly, right here, we have a cis arrangement there. Whereas you might have a trans arrangement. And that would alter the shape of the chain. So it would appear that, so there's our trans fatty acid. Right? So when you talk about cis and trans isomers, probably the easiest thing to remember here would be your fatty acids, because we've all heard about how trans fatty acids are bad for you. You don't want trans fats in your food, right? Well, cis fatty acids, this cis arrangement around a double bond, around an unsaturated fat, is sort of the natural double bond arrangement. All the naturally occurring fats all the naturally occurring unsaturated fats have cis arrangements around the double bonds. You only find trans arrangements, trans isomers around those double bonds when you do hydrogenation. So in hydrogenation, they actually take a unsaturated fat and break it down and flood it with hydrogen to try and turn it into a saturated fat. So, for instance, hydrogenated soybean oil, most plant fats, a large number of plant fats are going to have unsaturated oils in them, unsaturated fats. But plant fats are a lot cheaper than animal fats. The problem being that unsaturated fats are liquid, whereas saturated fats are solid. So if you're trying to make your tasty candy bar and put some tasty fat in there, and you don't want it to be a, liquid bowl of candy bar, you might want to try and convert a plant fat, which is nice and cheap, into a solid saturated fat. So you break it up, flood it with hydrogen, and try and convert it into a saturated fat. Byproducts of hydrogenation are unsaturated fats that are trans fats. And for some reason, all of the digestive capability in your body is geared towards cis fats. So for some reason, the trans double bond in unsaturated fats, artificially produced fats there, is very poorly digested by your body, and it can actually contribute more to disease, which sucks. Okay, so, good God, I went way ahead of myself. So single bonds rotate, the carbon-carbon single bonds can rotate independently of each other in opposite directions, but once you form a double bond, you form that axial. We no longer rotate. And so cis-trans is all about arrangement of atoms around that axial, around that double bond. When you have electronegative atoms in there, it can govern polarity, like our big example with oxygen. A cis isomer, where oxygen is on the same side, applies a polar side to that fat, or to that organic compound. Whereas if your oxygens are in trans position around that double bond, you won't have that same kind of polarity. So, in organic chemistry, we often use R as a shorthand for whatever, right? Because these organic compounds tend to be very large. We don't want to try and draw out every time the whole entire thing. So if we're focusing on one thing, we can just put R, the R group, or the rest of the whole thing. I like to think of it that way. R, the rest of it. So anyway, this is just illustrating cis-trans with simple R groups. So they're not even implying electronegativity or even what kind of compound this is. It's just some sort of organic compound, and here is a cis isomer where the R groups are on the same side, and here is a trans isomer where we have an R group on one side, and it seems to transmit across trans to the other side. So the trans isomer. Here's an actual compound, cis-2-butene and trans-2-butene. Probably one of the simplest organic compounds you could get that shows cis-trans isomerity. Isomerity? Whatever. Works for me. All right, and then here's my example with fatty acids. Cis and trans. On to the third type of isomer, which is a little more difficult to talk about, but basically we're talking about enantiomers. This works for odd-numbered carbon compounds. And they have to be odd-numbered because an enantiomer isomer is, or an enantiomer, is basically where you have two compounds that are mirror images of each other. So again, only occurs when you have an odd number of carbons. So here's a carbon double bonded to an oxygen and then single bonded to an OH. Let's see. So we have a carbon here. That COOH implies this. And then we have a carbon here. And then we have a carbon back here. And then we have hydrogens coming off. So hydrogen, and then we have coming off of there an NH2. So this is a amino acid there. So we have 1, 2, 3 carbons. So odd numbered. And this is just sort of showing you the depth of it, right? So because of that bond angling, right? This is all single bonds. So we have that tetrahedral shape, right? The three-sided pyramid. So there's our tetrahedron. And so we'll call this CH3 here being away. So it's sort of like back left, and then over here it's back right. And then this one sort of coming to the left, this NH2 here coming to the left, this NH2 coming to the right. So we change sides. We're mirrored. The enantiomers are mirrors. And they'll often use L and D to differentiate the two enantiomers. D-alanine and L-alanine. These are the two enantiomers. Okay, so a fun fact for enantiomers, or for isomers, excuse me, would be with regards to your ability to see. Human vision. I don't know about bug vision, but certainly with human vision, you have a protein down by your little eyeball sensors where light is detected called rhodopsin. So it's in your eyes. It's at the little cell that detects light, and basically it's a cis-trans isomer. Light causes the cis state to immediately reform into the trans state. And so when light impacts rhodopsin, the cis-rhodopsin rearranges into trans-rhodopsin. And when it does that, it actually triggers the nerve that says, hey, light here, light, 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 light. And it'll continue triggering light until a little chemical reaction occurs that actually rearranges it back, has to reset it. So this happens like millions of times per second, which is pretty amazing. That rhodopsin, whenever light hits it, is converted to its trans state. And then that triggers detection of light, and then it's immediately reset. Because if it wasn't immediately reset back to the cis state, then you'd get, bright white light. You wouldn't be able to see areas where there are light and no light. You wouldn't see any shadows and stuff. So, pretty cool. Light causing a conversion from the cis state to the trans state triggers your ability to actually sense light. I thought that was awesome. Might be an extra credit or even a good example of cis-trans. So now my cis-trans examples include my little fatty acids, cis and trans fatty acids, my little butane, my little example with oxygen, and rhodopsin. So you got all sorts of cis-trans isomer examples. All right, on to properties of organic compounds. So I keep talking about how different things on organic compounds will change their properties, add polarity, what have you. So let's talk about how that actually comes about, how these changes occur, and we have actually some common atoms or compounds that you can add to an organic compound that will confer very specific properties. So we can do combinations of these little compounds. We call them functional groups. So if you add functional groups to an organic compound, you will give it the properties of that functional group. So different combinations of these functional groups will confer different properties. These functional groups each have their own specific properties. So they change how we classify them, and they change the properties. So let's go through some functional groups. There are 59 important ones. So let's go ahead and just get started. The first off is a simple one, the hydroxyl, or the OH. We also always have a little single line here implying that it bonds there, right? So remember, oxygen being atomic number 8 can form 2 single bonds. So an OH already has 1 single bond. So this is where it attaches to my organic compound. OH means that a compound is an alcohol. Drinky, drinky. So you add an OH to an organic compound, and now you've got an alcohol, right? So methanol is where you have a single carbon with an OH and I think 2 hydrogens. And ethanol is where you've got 2 carbons. Ethanol being the one you can drink. I don't recommend you drink methanol unless you like going blind and having your liver fall apart. So, if you're going to drink alcohol, stick to ethanol. I had a friend in college who, since ethanol is the alcohol in, everything you drink, you can abbreviate it ETOH. ET for ethane and OH for the alcohol, so ethanol. She used to say, "Hey, John, you want to go out and get some ETOH?" So that was our nerdy talk for grab a couple beers. I thought that was pretty great. So I have taught it to you, and now you can say it and impress all the people to whom you are attracted. All right, amino groups. You'll never guess what sort of compound we find an amino group on. Oh, NH2. The amino is NH2. And there we go, RNHH. We find it on amino acids. It's responsible for the amino in the amino acid. Gasp. I know, such a surprise. Phosphate groups. Phosphates are pretty fascinating. See, a phosphate is highly electronegative, or not the phosphate. It has 3 oxygens on it, which are highly electronegative. And that confers essentially a negative charge to the phosphate. OPO3 with a 3 negative charge. So phosphate is actually heavily charged. And we actually find phosphate in ATP, the energy for life. The currency for energy in the cell is ATP, or adenosine triphosphate. So how that works is you'll have some phosphate groups, 3 of them, attached to each other. And each one is negatively charged. And so they are trying to push away from each other. And because of this, you have an extremely unstable compound. And that is actually why ATP is good for energy, because of those 3 phosphates trying to push against each other, creating an unstable compound. You have a lot of sort of energy locked up. The more unstable something is, the more energy you can produce when you break it. Think, dynamite is highly unstable. And when you blow it up, you get a lot of energy. So ATP, highly unstable because of those charges. So when you break ATP up, you get a bunch of energy from breaking the bonds. Phosphate is important in nucleic acids, right? So when you talk about DNA, right, you'll talk about that double helix, right? So there's the helix. Ooh. Anyway, so when you get down to it, DNA is basically a twisted ladder. Like, here's a flattened out ladder, right? And if you get real close, you have a sugar and then a phosphate holding together DNA. So these are my phosphate groups, and these are my sugars, right? And so that ladder is actually made of sugars and phosphates. So phosphate forms, so you have these little sugars here. Sure, why not? And joining each sugar is a phosphate. So we'll throw our phosphates in here. Boom. They actually call this the sugar phosphate backbone. Sugar phosphate. Sugar phosphate. So the sugar phosphate backbone. That's what gives DNA its strong backbone. So pretty darn cool. Phosphate is involved in respiration, because respiration is how we make the ATPs. So there's a phosphate. Boom, carboxyl groups. The carboxyl group is an acid. It confers acidic properties. So it's a carbon, right? A carbon double bonded to an oxygen and single bonded to an oxygen which is bonded to the hydrogen. I cannot imply that enough, because when you see it written out, most abbreviations will just write COOH, put the dash there. And if you were to just like diagram it out with the chemistry set or on paper, you might think it looks like this, right? Because it just says COOH. And if you do that, well, you've got a problem here, because carbon can make 4 single bonds. So This is not a full valence. And our functional groups always have only one empty single bonding site to connect up to the rest of it. So it is this compound here where we have a double bond to an oxygen and then a single bond to an OH. And that is the carboxyl group. So COOH, it is acidic, sometimes called the carboxylic acid. And so basically it gives us the acid part of any kind of molecule that's organic where we say acid. I can't say that universally, but as far as biological molecules go, fatty acids have a carboxyl. Amino acids have a carboxyl. Nucleic acids have a carboxyl. They're all through it. And no, we're not going to go through all 59 because this is biology. You can learn that in organic chemistry. So torture yourself there. Here we're just Bio 1. So you do need to know the previous 4 I went through and the basics about them, their properties. And there is a figure. It used to be 4.9 in your book if you're using the book. Otherwise, you could Google common amino, or not common amino acids. You could Google functional groups and just go over to images. So, that's it. We're done, people. Finally. So, yeah, have a good time. Oh, crap. How do I end the recording? Control-Shift space. This is going to get cut.